Ohayo gozaimasu. My name is Yating. I don't have Japanese name, but however, I have a Latinx name, Giuliano. And, hello. And I'm an assistant professor from Taipei University. And today I'm going to share with you my recent project. And the title is Exploring Hypothalamic Influence on Metabolic Dysregulation. There's a saying that it takes a whole village to raise a child. And for me, I believe it also takes a whole village to maintain the energy homeostasis. The energy homeostasis transfers our food intake into the energy we are going to use for everyday daily life. For example, I'm standing right here. It takes a lot of energy of me. So talk about energy homeostasis. There's a lot of different systems and organs involved, which including the digestive system, the pancreas, liver, other tissues, and also the muscles. However, our central nervous system serves as a very crucial role to coordinate the whole system and maintain the homeostasis. And in our central nervous system, one area particularly is important called hypothalamus. Hypothalamus is located in the medial basal part of our central nervous system. It's located right here. And hypothalamus is divided in a lot of different areas, which including the paraventricular nucleus, VNH, LH, and also the arcuate nucleus. And hypothalamus will receive nutrient signals like insulin or leptin, also including like glucose, fatty acid, or amino acid signals. So the neurons in the hypothalamus will innervate the nerve terminal to the hidden brain, an area called nucleus tract surrender. So in this neuron in this area will send signal to the peripheral organs through vagus nerve and sympathetic nerve system. So by activation of this neuron system, our central nervous system will regulate the hormone secretion, like in the pancreas, the insulin or glucagon secretion, or the hepatic glucose production to regulate the glucose homeostasis, and also for the feeding behavior and energy expenditure, including the brown adipose tissue thermogenesis or white adipose tissue browning. So let's take closely to the hypothalamus. This slide shows the neural circuits involving the hypothalamus. And one area is particularly important is arcuate nucleus, short as ARC. So due to its special structure, the insulin can pass through the blood-brain barrier right here and binds to its receptor located inside the nucleus, arcuate nucleus. So in this area, they contain two types of very specific neurons. One type of neuron expresses NPY and AGRP. By activating this type of neuron, this will increase the feeding behavior but decrease the energy expenditure. However, the other type of neuron expresses PUNC. So through the post-translational process, a peptide called alpha MSH will be released. So alpha MSH will reduce the feeding behavior, on the other hand, increase the energy expenditure. So by binding with insulin receptor, here insulin will activate the PUNC neuron activity, however, inhibit the NPY and AGRP neuron activity. So at the end, insulin will inhibit our feeding behavior, but increase the energy expenditure. And the neuron from arcuate nucleus will also send their nerve terminal in the neighborhood area, which including PBN, BMN, and also the LH. There's a little bit complex neuron circuits involved, but in the end, they will send a nerve terminal to the NTS in the hindbrain. So through the vagus nerve and sympathetic nerve, the signal will regulate the behavior and the metabolic effect of energy balance. There's another neural area called VTA and the nucleus accumbens. They are involved into the reward system and the which is act as the decision when if we like the food or if we want more of the food. So talk about metabolic disorder, we will think about insulin resistance, right? But insulin resistance not only happen in the peripheral organs. Insulin resistance can also happen in the central nerve brain. It is called central insulin resistance, or also called hypothalamic insulin resistance. So insulin resistance can happen in the arcuate nucleus because this is the location where the insulin receptors are. So by trigger the insulin resistance in the arcuate nucleus, it will lead to the metabolic disorder like obesity, metabolic syndrome, type 2 diabetes, and also the cognitive disorders. A lot of different factors, which including the inflammation, alter the blood-brain barrier, or ER stress, will trigger the central insulin resistance. One molecule in our lab we are particularly interested is neuropeptide FF, short as NPFF. NPFF is highly expressed in the hypothalamic and has been reported to involve several hypothalamic functions, including the role in stress response, mood disorder, glucose homeostasis, thermogenesis, and also the energy balance. It has been demonstrated that NPFF has two receptors, NPFR1 and NPFR2 receptor. Both of them are G-protein coupled receptor. But recent publications indicate that NPFR2 is the receptor responsible for the function of NPFF in energy in modulate of energy homeostasis. So in my study, I will focus on NPFR2 receptors. So first, I want to know if the NPFR2 receptor co-express with insulin receptor in the hypothalamus arcuate nucleus. This is a slice from mouse brain, and this is the cerebroventricular, and this is the area of arcuate nucleus. So by RNA scope, we stand the expression of NPFR2 in green or green fluorescent signal, and we also stand the insulin receptor indicate in red or red fluorescent signal. So by merge of these two signals, we can find that the NPFR2 receptor and insulin receptor can co-express in the same neuron. And by analyze the result, we found that among all the NPFR2 receptors in the arcuate nucleus, there's about 20% of them co-express with insulin receptor. On the other hand, among all the insulin receptors in the arcuate nucleus, there's about 18% of them co-express with NPFR2. So our goal is to investigate if the NPFF acts through NPFR2 in the arcuate nucleus to send signal to the hipbrain and regulate the central insulin cascade and the downstream metabolic disorders. So my study will start with the in vitro study. So we're using the mouse hypothalamus cell line to see if the NPFF will regulate the insulin downstream cascade. So this image shows the insulin signaling pathway. Insulin will bind to insulin receptor and phosphorylate IIS, and then activate the PI3K, which further phosphorylate AKT and leads to a transcription factor called FOXO1. The transcription factor FOXO1 will translocate from nucleus to cytoplasma and be degraded by ubiquitinase. And this will eventually lead to the reduction. The insulin will lead to increase of POMC and CART gene expression, but on the other hand, reduction the HRP gene expression. So in this hypothalamus cell line, we treat the cell with insulin. The insulin will increase the AKT phosphorylation. However, when we treat the cell with insulin and NPFF, we found that the treatment of NPFF significantly reduced the insulin activate AKT phosphorylation. By using the immunofluorescence staining, we observe the FOXO1 translocation. So under basal condition, the FOXO1 was located mostly in the nucleus. However, after treating with insulin, we found that the FOXO1 translocated from nucleus to cytoplasma. And the effect, the translocation is reversed by we co-treat the cell with MPFF and the insulin. We also measure the transcriptional level of PUMC. So we found that the insulin can upregulate the PUMC transcriptional level, however the effect is reversed by co-treat with MPFF. So we want to confirm the effect of MPFF on insulin cascade is mediated through MPFF-R2 receptor. So we performed the same study, but this time we used MPFF-R2 agonist DMPA to replace MPFF. So we found that the very similar result that the insulin activate AKT signal can be inhibited by the pretreatment of DMPA. And also, to confirm the effect of MPFF is mediated through MPFF-R2. So we transfect the cell with MPFF-R2 siRNA for 72 hours. So we found that after silence the MPFF-R2 expression, after silence that the effect of MPFF on insulin activate AKT phosphorylation is reversed. So in our in vitro study, we demonstrate that MPFF can act on MPFF-R2 receptor and inhibit the central insulin cascade. So further, we want to find is this signal pathway will affect the metabolic disorders. So we move our study from in vitro to in vivo. So we use animal models. In this animal model, we overexpress MPFF-R2 directly inside the brain into the arcuate nucleus. So we inject lentivirus packaged MPFF-R2 plasmid into the arcuate nucleus. So this slide shows that this is the area of ARC. There's a significant fluorescent signal detected right here, indicate that we can overexpress the plasmid in the arcuate nucleus. And by measuring the transcription level of MPFF-R2, we also found that there's a significant increase of MPFF-R2. So 2 weeks after surgery, we start to establish metabolic disorders models to type 2 diabetes models. So by expose the mice to high-fat diet for 12 weeks, and at week 6, we inject the mice with STD to induce the type 2 diabetes models. We measure a lot of different metabolic parameters, but here today I'm only show part of them. First is the feeding behavior. So we measure the weekly food intake. We found that compared to control group, the arcuate nucleus overexpression MPFR2 will increase the feeding behavior, and this was supported by the gene expression in the hypothalamus, which we found that the reduction of PUMC and CART gene expression, both of two were the energetic peptide, which will reduce the feeding behavior. On the other hand, we found that the overexpression of MPY, the overexpression of MPY will lead to the increase of feeding behavior. So we also analyze the glucose homeostasis. So here we can find that the circulating insulin level was significantly higher in these overexpression mice compared to control group. And also the hormone IR index indicate the insulin resistance is also higher compared to control group. So we want to analyze the insulin resistance. So we use the insulin tolerance test. So by injecting the insulin into the mice, the blood glucose level will decrease. But however, we found that in these overexpression mice, they have higher blood glucose level compared to control group. So this is the result of area on the curve also shows significant insulin intolerance in these overexpression mice compared to control group. So further, we collect the tissue, the peripheral tissue like liver and white adipocyte. So we analyze the lipid profile in these tissues. So this is the liver slice and stained by HE staining. And the above is the control group, and the below is MPFR2 overexpression group. So we can find that the hepatic cirrhosis was significantly higher in the overexpression mice compared to control group. And we also observed the lipid size of Y-active tissue in the overexpression mice. We can find that the lipid size was significantly larger compared to control group. However, when we study the pro-inflammatory cytokine TNF-alpha in white adipose tissue, there is no significant difference. So similar strategy, but this time we use the obesity models. So after overexpression MPFR2 in the arcuate nucleus, we feed the mice with high-beta diet to induce obese mice. And we found that similar result in feeding behavior, that these mice show increased food intake but a reduction of heart gene expressions. So let's take a look at glucose homeostasis. So similar result, we found that the overexpression of these mice have a higher circulating insulin level and also the higher HOMA-IR index. So in this model, we test both insulin tolerance and glucose tolerance by IV injection of insulin and glucose. And we found that the overexpression mice has severe insulin intolerance and severe glucose intolerance compared to control mice. Again, we analyze the difference changes in the peripheral tissues, but this time we collect the brown adipose tissue along with liver and white adipocyte. So in the brown adipose tissue, we stand a protein called UCP1. This protein is the marker of thermogenesis. By increasing this protein, this will increase the browning of the brown adipose tissue. And we found that the overexpression mice has significantly less UCP1 in the brown adipose tissue compared to control group. And similar finding compared to type 2 diabetes model, these overexpression mice also have a significant large lipid size in white adipose tissue compared to control group. But this time we found that there is a significant increase of pro-inflammatory cytokine TNF-alpha in white adipose tissue compared to control group. And in the liver, we can, it is obviously that the high-beta diet will induce the liver cirrhosis, and here we can find that the hepatic cirrhosis was severe in the overexpression mice compared to control group. So further, we want to investigate the more detailed molecular mechanism. So we collect the arcuate nucleus for analyze the gene profile by RNA sequence. So we identified 15-6 gene significant changes in these overexpression mice. Some of them was increased in these mice, and some of them was decreased in these mice. And we further analyzed the result using Go analysis, and we identified several pathways. And some of the pathway was related to our project, which including the hormone secretion, hormone transport, glucose homeostasis, regulation of hormone secretion, and also regulation of insulin secretion. So we also confirmed that 2 of the genes analyzed by the next generation sequence by real-time PCR. So we found that a similar result, that there's increase of PDB3B in the overexpression mice, but a decrease of RBP4 in the overexpression mice. These 2 genes was all related to the regulation of insulin secretion. So in our lab, we are now focused on the study, the detail of molecular mechanism of how MPFR2 impact on the central insulin resistance and leads to the metabolic disorders. So that is still in the process. So I'm going to summarize our findings. So through the in vivo to in vitro study, we demonstrate that in the hypothalamus arcuate nucleus, MPFF will bind to MPFR2 receptor, inhibit the insulin cascade. So by inhibiting this insulin cascade, this will lead to central insulin resistance. And the central insulin resistance had a adverse effect on metabolic disorders like type 2 diabetes and obesity in mice. So this is my last slide. So I'm going to acknowledgement of my mentor, Professor Jinzhong Chen, and my husband, Dr. Tohoku. He's forever my partner, and also my student involved in this study, Wu Guanxuan, Zhang Jielan, Zhang Jiezhu, You Xia, and Cai Shiqi. And I'm also very glad to have this opportunity, and thanks for Taipei University and Tohoku University have me here to present my recent project to you. And last but not least, I'm always thanks for the animal sacrifice in these studies. So this is my talk today. I'm open for possible questions or comments. Thank you.